Welcome back to the lab. We've learned how to manipulate genes and screen for success, but today we're going a step further, focusing on the fundamental units themselves cells. We're diving into cell cloning, the powerful technique that lets us create perfect copies of cells, essential for everything from gene editing to disease modeling. And we're back. At its core, cell cloning is a crucial laboratory technique used to generate genetically identical cell populations from a single cell. Think of it as creating an army of identical cells, all originating from one founding cell. This process is absolutely essential for gene editing, disease modeling, and therapeutic research. Why is this so important? When you're working with cells in the lab, you need a uniform population to ensure your experiments are reliable and reproducible. If you have a mixed bag of cells, your results can be inconsistent. Cell cloning provides that consistency, ensuring every cell in your experiment behaves the same way. The basic principle involves isolating a single cell and then allowing it to multiply and form a colony, a group of cells all derived from that one original cell. This uniform population is invaluable for detailed studies. Now, let's talk about how this magic happens on the bench. Cell cloning can be done manually or with the help of automation, and both have their place in the lab. Manual methods. Traditional approaches include limiting dilution and manual colony picking. While these methods are straightforward, they can be time-consuming and sometimes lack absolute proof that your colony came from just one cell. Newer manual methods sometimes use tools like cloning cylinders or tissue gels to make the process quicker and reduce handling errors. Automated systems. This is where modern technology truly shines. Devices like Iota Sciences IsoCell, Selenium Cell-in-One, and Cytina dispensers automate the isolation and seeding of single cells. These automated systems dramatically increase throughput, making them perfect for large-scale experiments and provide documented proof of clonality. This means you have a traceable record that your cell line truly originated from a single cell, which is critical for regulatory compliance in therapeutic applications. Regardless of the method, the core steps involve preparing your culture plates, dissociating your cells, isolating single cells, seeding them into wells, and then monitoring their clonal expansion. Special protocols for sensitive cells, like human pluripotent stem cells, HPSCs, often use techniques like FACS, fluorescence-activated cell sorting, or microfluidic cell sorting, along with specific anti-apoptotic supplements to boost cell survival and efficiency. If you recall, we discussed the power of CRISPR in our gene cloning episode, and FACS is often used hand-in-hand with CRISPR to isolate successfully edited cells. Even with advanced tools, cell cloning can be challenging, but a few key optimization strategies and troubleshooting tips can dramatically improve your success rate. Low cloning efficiency. This is a common hurdle. To overcome it, use anti-apoptotic supplements like the CEPT cocktail, practice gentle cell handling during isolation, and ensure your media is perfectly optimized. Contamination, a constant threat in cell culture. Strict aseptic technique is paramount, and regular media changes are essential to prevent contamination. Multiple colonies per well. To ensure you have a truly monoclonal population, you need to validate it. Visual inspection helps, but microfluidic devices offer precise isolation to ensure only one cell is deposited per well. Slow growth. If your cells aren't proliferating well, optimize your media and supplements, and ensure proper plate coating using materials like Geltrex or laminin. After successful clonal expansion, you'll need to screen and validate your colonies. This often involves techniques like PCR and DNA sequencing to confirm genetic identity and detect any mutations. For larger fragments or protein expression, you might even use gel electrophoresis to analyze fragment size or Western blots to assess protein production. We've covered all these verification techniques in other episodes, so check those out if you need a refresher. So why go through all this effort? The applications of cell cloning are truly transformative. Gene editing. Cell cloning is absolutely essential for generating stable edited cell lines after techniques like CRISPR-Cas9. This allows researchers to create precise genetic modifications and study their effects. Disease modeling and drug testing. By creating uniform cell populations, scientists can build highly reliable models of diseases in a dish, allowing for accurate drug screening and testing. Therapeutic protein production. Cell cloning enables the creation of monoclonal cell lines that are specifically engineered to produce therapeutic proteins, such as antibodies or vaccines, critical for biomanufacturing. Regenerative medicine. The ability to expand specific cell populations from a single cell is foundational for developing cell-based therapies for various diseases. Cell cloning, whether manual or automated, is a fundamental technique that underpins much of modern biological research and biotechnology. Advances in automation and supportive reagents continue to make this process faster, more reliable, and accessible for a wide range of scientific and medical applications. From creating perfect copies for research to revolutionizing gene editing and drug discovery, cell cloning truly is a cornerstone of modern biotech. It's a testament to how meticulous work at the cellular level can lead to groundbreaking scientific advancements. If you're fascinated by the building blocks of life and the incredible ways we can manipulate them, hit that subscribe button and ring the notification bell. Our How to Biotech Lab playlist is your go-to resource for understanding these complex techniques. Join us next time as we explore another exciting frontier in the world of biotechnology.